All righty, so it's my pleasure to introduce now the Cancer Biology and Genetics section. And it's Dr. Harada, who is supposed to be my co-host, unfortunately isn't here. So I will do this all on my own, so that hopefully I hope I can do it. Thank you. Thank you for the vote of support, Nick. I appreciate it. Okay, so our first speaker is somebody who I think is known to everybody, Sir David Lane, and he's going to be talking about the p53 pathway in zebrafish. I should also say just before we start that we're going to be having lunch together, and the organizers of the meeting have made it so that 3 speakers from each, there's 30 total speakers here, they'll each be sitting at different tables. But I would also like to encourage at lunch that that we mix completely. So this would be Asian and non-Asian groups. I think we want to encourage us to interact. So please don't hang out with your friends, but actually just your close friends, Nick. Okay, Dr. Lane.
Okay.
Oh, you're all set.
I'm okay. Thanks very much. Well, thanks very much, and it's really, really very nice to be here. It's great for me to come to a meeting where there is no time change. So Singapore and China were on the same zone, so I'm kind of awake and wired while the rest of you are just about to fall asleep, but never mind. Anyway, the other thing I want to say, most important really, is the bar. So the bar is great, and if you don't get to meet the person you want to meet at lunch, I can assure you that a lot of the speakers will be in the bar tonight, and very happy to talk to you then. Okay, so I'm going to try and stick to time. If I can, yeah. Okay, so the 2 people who are here from my lab and who are having a poster this afternoon also on what I'm talking about are Guo Lin, and this is him at our Chinese New Year dinner. So those people who come to my lab get nice Chinese New Year dinner, and this is Ho Peng, his colleague, and they'll be around and happy to talk to you and explain what we really did as opposed to the boss's version of what happened. So the story is all about p53 and p53 stability. So the way that p53 is regulated in cells is complex, but one of the key ways it's regulated is by altering its half-life. And the protein is constantly synthesized and constantly broken down, and that happens through a very efficient ubiquitin E3 ligase system that consists of the protein MDM2, often working in partnership as a heterodimer with MDM4. And this regulation of half-life is a critical way that the pathway is controlled, and many of the controls on the pathway actually act on this E3 ligase itself. And this is a theme that's emerging in many areas of protein control, that the E3 ligase is a critical point of control. And so many of the signals that activate the p53 response do so by affecting the function of this E3 ligase. And there are different ways this can be achieved, and one very powerful route is by the physical binding of proteins to the MDM2 protein. And examples of proteins that bind to MDM2 and inhibit its function are the small, highly charged ARF protein that's found in mammalian systems and is a very critical tumor suppressor, but is absent in zebrafish, at least as far as we can tell. And then very importantly and more recently, the realization that a large number of ribosomal proteins very specifically interact with the acidic region of this protein and to regulate its function. There's a lot of papers now showing that a whole variety of human syndromes that involve mutations in ribosomal protein genes affect this axis. The Diamond-Blackfan syndrome and others are affected by this. And this is a very critical control pathway that regulates p53. Now, another pathway that regulates p53 is the DNA damage pathway, and that also acts on MDM2 through ATM-dependent phosphorylation. And finally, we know that now that a very large number of different developmental defects are detected by this axis. And in zebrafish, a very large number of mutants, part of their phenotype is achieved by affecting this axis. And originally this was seen with morpholinos, but more recently it's been very thoroughly done now with a whole series of real mutations showing that these mutations talk to this axis. Now, in the mouse field, we're very fortunate now that we've got to a higher level of resolution, and there are 2 very important knock-in mutants, point mutants in mouse now that distinguish these pathways. So there's a point mutation in the zinc finger of MDM2 that blocks the binding of the ribosomal proteins. And those mice appear normal, but they have profound defects in metabolic signaling. So when you starve them, they don't know they've been starved, and they don't alter their metabolism. And they're very sensitive to MYC-induced tumorigenesis. So MYC is actually signaling to the p53 axis through two different pathways that interact with MDM2. In the case of DNA damage, not yet published, but there's a single point mutation in one of the ATM phosphorylation sites that, when knocked into a mouse, makes the mouse extraordinarily resistant to ionizing radiation. So the emerging picture is that MDM2 is receiving multiple signals, and it's doing so through regulating its activity as an E3 ligase, and that can be achieved in very specific ways. And I think the further dissection of this is going to be extremely valuable and important in many areas. And we don't know yet about these defects. I mean, clearly we need to try and develop this system so that we have more point mutations in MDM2 and try and see, does development signal through a damage-type pathway or through a ribosome biogenesis pathway or through some other mechanisms to this axis. So the push in our field, rather like Phil described last night, has been to develop drugs based on this pathway, and most progress there has been made in looking at this particular axis. That is the case of tumors where p53 is wild type. Now, in about 60% of human tumors, p53 is mutant, and actually as the clonal sequencing techniques are coming through and projects are getting better, the frequency of p53 mutation in human cancers is rising. And from Vogelstein's analysis, we can now say it really is the most frequent mutation that he's detected in whole cancer genome sequencing. So point mutation in p53 is really a very striking characteristic of human cancer. But about 30 to 40% of tumors don't have mutations in the coding gene. And in many of those, we can find systems that damp down the p53 pathway. So most clearly, of course, in the case of cervical cancer and some other cancer types now, the HPV-E6 protein blocks p53 function by directly degrading it through an MDM2 independent pathway. And then in many other tumors, we find overexpression of MDM2 or a suppression of ARF. All of these stop this signaling pathway so that the tumors can grow even in the presence of wild-type p53 because the cells are not detecting the signal. And as I'll describe later, in fact, one of the problems in our field is this system's immensely sensitive, and routine tissue culture is a stress to this pathway. So most of the experiments that have been done on the p53 pathway in tissue culture are flawed, really, because the system is constantly signaling, and the only way the cells can grow is by switching off the signal. Now, this pathway is the one that has been proved susceptible to drug development, and the idea has been to develop small molecule inhibitors of the p53 MDM2 interaction to overcome this block. So this upstream block can be overcome by interfering at this point. And these molecules should activate the p53 pathway, but don't require a stress signal. So they shouldn't be toxic or genotoxic. Their activity should be entirely due to interruption of the signal. And here's the molecule that's currently in clinical trial. There's now a number of these molecules, and the story is getting very interesting. So from one pharmaceutical having one molecule, we now have about 5 pharmaceuticals companies having molecules. So this was considered an undruggable target because it's a protein-protein interaction. But one of the great truths, I think, in life is that once somebody gets one, everybody else seems to find it very easy to get one. And so now it's not an undruggable target. In fact, it's an overdrugged target. And the other nasty thing that's emerged from the woodwork is that, and I heard it last night, though the binding site here is hugely conserved in evolution. In fact, we showed even in Placozoa that it has a similar molecular interaction. The detail, the precise detail matters. And many of the drugs that are currently being tried look fabulous in xenografts. They completely wipe out the human tumor cells, and the mouse has no toxicity at all. But very recently it's been found that's because these drugs only bind to human and not mouse MDM2. So nutlin cross-reacts very nicely across species. But as you get to picomolar inhibitors, they start to get species-specific. So you've got to be very careful and measure everything. And well, anyway, no, that's not fair. I'll do that in the bar, but you have to be careful about this stuff. Okay, so the other approach that we've started, and Greg Verdeen and his colleagues have pioneered, is really very exciting, and that's the use of this peptide stapling method, where you could take a peptide and turn it into a drug by putting in a chemical crosslink. And these really do work very, very well, and I think give a very promising prospect for many, many pathways that we're investigating that will have drug-like molecules that we can use to study them. So I'm not going to talk about those things anymore. I just thought it was fair to give an introduction to where the field was. And what I want to focus on today is to talk about the stability of mutant p53 and why that's important. So if we look in mice models, we find that we get tumors in mice simply by complete loss of function of p53, complete deletion. But of course, in humans, we don't have that situation very frequently. Most frequently, we have point mutation. So we find the full-length proteins expressed in the tumor, and it's usually got a single amino acid change, a single nucleotide change, typically in this DNA binding region, though there are mutations in the oligomerization domain. And as a slight diversion, there's 90,000 people living in Brazil with a point mutation in their oligomerization domain, and they have a cancer susceptibility that's very specific. But the majority of the mutations are in this DNA binding region. And they're expressed frequently at high levels, and it's that expression that I want to particularly talk about. So what are the consequences of mutation in p53 as opposed to loss of expression? And we can imagine a number of consequences. So certainly we lose wild-type activity. We've only got, we've lost the wild type, we've lost its function, and it's very important to recognize that p53 is haploinsufficient. So the cell knows whether it's got 1 or 2 copies of the p53 gene in functional assays in a cell-autonomous way. So simply losing one wild-type allele is a significant biological event. And then in many cases, these mutant proteins can heteroligomerize with the wild-type allele if it's present, and you can get a dominant negative inhibition of wild-type p53. But what's excited a lot of interest recently is the realization that many of these point mutants have a gain of function. That is, they do something even in a null background. So the mutant protein has a new biological activity that can promote invasion and metastasis. And often that's dependent on expressing high levels of the mutant protein. So what I'm going to talk about for the rest of the talk is this phenomena, this accumulation of mutant p53 that can happen in human tumors. And you can see it very clearly by just immunohistochemical staining of human tumors. And there's an enormous number of reports of this. The staining patterns are quite heterogeneous and various. Not every tumor cell stains, and there's a correlation between staining and mutation, but it's not absolutely clear, as we'll see. So what's been important here is the proof that this expression of mutant protein is biologically significant, and this really came from 2 papers that were published a number of years ago now, where they compared the tumor phenotype in mice that lacked p53 or mice that were expressing a mutant p53 allele alone, no wild-type allele. So here we're not looking at dominant negative, we're looking at potential gain of function. And what was found in these papers is that the big effect of this expression of mutant was that the mice now got carcinomas, and these were invasive and 11 minutes, and metastatic. So this is very important. This suggested that in those human tumors that are expressing mutant p53, that mutant p53 might be driving the tumor in this way. And there's a lot of work now to support this, and Karen Woosden and others particularly have shown very clearly that in tissue culture models and in animal models, you can show that expression of mutant p53 actually drives invasion and metastasis. And it's partly through inhibiting the functions of the p53 family members, p63 and p73. So these tumors arise, and you can show that they're important. And the critical issue is that what we have and others have shown is that this accumulation of mutant p53 that occurs in these tumors is a tumor-specific event. So if we look in the normal tissues of either mice that are mutant over null or mutant over mutant fish, we don't see this accumulation in normal tissue, and this is the main thing I'm going to talk about. So this gets more interesting when you talk to pathologists who've been looking in detail at preneoplasia in man. So this is work from Chris Crum and others, and he's been looking at the origin of ovarian cancer, and particularly looking at the fallopian tube, where he believes the precursors for ovarian cancer arise. And what you see, and this is a fallopian tube from a woman who has a BRCA mutation, you see these areas of extremely intense p53 staining, and these are called p53 signatures. And you see the same thing in our skin. So everybody in this room will have these little foci of p53 positive cells in their skin, in their sun-exposed skin. And you can see here these intense staining clones in the epidermis. And these range from lesions that look completely normal morphologically to what look like very interesting pre-neoplasia neoplasia developments, where you can see regions where the same clone is present, and in fact by sequencing the same mutation, and yet in some areas it looks like relatively normal tissue, and then it looks like an early beginnings of cancer here. So what's causing this phenomena, and what's its relationship to the control of p53 protein stability, which we've talked about? What's the role of MDM2? What is this mutant protein doing? Is it biologically active? These are the kinds of questions that we want to get at. So we've been using the Zebrafish model to do this because I think Zebrafish has a number of advantages for us in looking at this particular system. And that's because we basically can reiterate and study all these steps very easily. And this all depends upon the availability of the p53 mutant Zebrafish that's been so generously distributed in the community. So the p53 mutant fish, these are, we can make them either, we don't have a null allele, so we can either make fish that are plus minus for this allele or homozygous mutant. And these fish develop Schwann cell tumors, and as Liz showed, in the presence of BRAF, they develop malignant melanomas. And we can use them as a model to study p53 stability, because we have an animal that's homozygous mutant. It's only able to make the mutant protein, so that allows us to study that particular state. In these fish, we were able to detect these isoforms of p53. And as you know, in the fish model, the role of the ribosomal gene in promoting tumors has been studied in a very interesting report saying that there's a different sort of state. So some of these tumors that have these ribosomal gene haploinsufficiency seem to be defective in synthesizing p53 protein at all. So this is just to remind you of the location of the point mutant allele that we've been using, and the presence of this delta allele of p53, which makes a very abundant message. So this was confusing in the field for a while, because not only is this message much more abundant than the message that encodes the full protein, but this messenger RNA's expression is totally dependent on full-length p53 function. So you only see this form of delta p53 where p53 is wild type, and that's because the promoter that drives the expression of this form is exquisitely p53 dependent. And the work we did initially was with Jin Rong, who's here at the meeting with his team, looking at this developmental mutants. And this is Jin Chen and Jin Rong, who are both here. And we published these papers a little while ago, basically showing that this deaf mutant had a defect in liver and gut development, that the mutation triggered the p53 response. So as I said at the beginning, it's one of these mutations that triggers the activity, and identified this as a p53 responsive gene. And showing essentially that p53 has a role in protecting development. So we've made this panel of monoclonal antibodies to p53, and the ones we use most now are ones against the N and C termini. And we can use these to stain for p53 in zebrafish and look at the localization and expression of the protein and its stability. And we can use these in Western blots as well. And you can see they're very clean antibodies if we get the conditions right, so we get almost no signal from an untreated fish, and we can get a very strong signal from a fish that's been exposed either to drug or ionizing radiation. And so you can study the expression of p53 using these antibodies, and you can see in different stages in development which cells are induced by drug to stabilize p53. And this is the background, very low background in the control animal. And you could show it here with ionizing radiation and with combinations of drugs. So from those early work, the conclusion was that the wild-type p53 is induced in fish, as it is in mice and man, by non-genotoxic drugs and by radiation. We can actually induce p53 in fish with this small molecule inhibitor, Nutlin, and the induction where we see p53 induced is very characteristic and cell type specific and developmental stage specific. So that set the scene then for looking at the expression of mutant p53. And in the original model, it was a very beautiful model. It says, well, the reason that mutant p53 accumulates is because it can't turn on its negative regulator, MDM2. MDM2 is a p53 responsive gene. You have this feedback loop where high levels of p53 turn on MDM2, and MDM2 then degrades p53, and that sort of sets the homeostasis of the system. But that turned out not, though it's a beautiful model, it's not right, or it's not completely right, as we'll see. And in fact, if you look at the mutant fish, untreated, they just look like the wild-type fish. They don't have any accumulation of p53, even though they're only able to express the mutant protein, and that mutant protein is defective in inducing MDM2. And we think this is because the MDM2 has a complex promoter, has two different promoter elements, one of which is p53 dependent, and one of which is p53 independent. So even in the absence of p53, you do produce a certain level of MDM2. And as I'll show, that's sufficient to control the level of p53, of mutant p53. So in the absence of stress, we suspect that the system is maintained not by p53-driven MDM2, but by MDM2 that's driven off this other promoter. And it's only when stress happens that you engage the feedback loop. But these fish are, just like the wild type, incredibly responsive in terms of p53 stabilization of the mutant protein now to drugs and radiation. And you can see here the ionizing radiation or drugs is very nice accumulation of p53. Now, the question then for us is, since the mutant protein doesn't accumulate in the normal tissue of these mutant-only fish, what happens in the tumors that they develop? And so you can very easily study that by just looking at fish tumors and staining them with our anti-p53 antibodies. And what you see in these tumors is extremely clear tumor-specific accumulation of p53. And it's very dramatic. These are two antibodies, one to the N, one to the C terminus. This is a control. So you can see this very, very clear accumulation. You can see it by Western blotting. Really massive levels of p53 are accumulating specifically in the tumor. And you can see it by closer examination of immunohistochemistry. And I mean, what's interesting, and I haven't got a lot of slides to show you because of time, but it just looks like the human tumor situation. So you see some tumors where every nucleus stains with p53, some where only some do. And yet we know now, we've simplified the situation, all of these tumors are only able to express this particular mutant. So it's a very clear illustration that this heterogeneity and tumor specificity is coming about because of some cellular event, and it's not directly related to the nature of the mutation itself. So what is controlling the level of mutant p53? How can we understand what's going on in the tumor and what's going on after radiation? So we can do morpholino knockdown of both MDM2 and MDM4, and in both cases, when we knock down the E3 ligase, we see the accumulation of p53 very dramatically. And we're just in the process now of making MDM2 and MDM4 zinc finger knockout fish that will allow us to study this in more detail. So mutant p53 is not stable in normal undamaged tissue. Staining is induced by damage and drugs in the same way as we see in the wild type. And both MDM2 and MDM4 are important in maintaining low levels of mutant p53 in the fish. And that's a morpholino result, but we hope soon to turn it into a pure genetic result. And therefore, mutant p53 staining in tumors represents a tumor-specific activation of p53 stability pathway. And I just want to pause there for a moment, just sort of consider that. So that means that in tumors, this potential protein that's driving invasion and metastasis is getting specifically stabilized. And actually, that same stability could be induced by drugs or damage. So when we give a patient chemotherapy, we may be stabilizing the expression of a potential oncoprotein, the mutant p53. So the implications of this is very significant, I think, for trying to understand what happens in response to therapy and the role of this particular mutant protein. So the feedback loop, the way that p53 is regulated by MDM2 and turns on MDM2, has been a real cornerstone of the p53 field, and it's very disturbing in a way, or it was at the time, to see these results where this particular homeostatic pathway didn't seem to be very important. But then we thought a bit more and said, it's one of the few times I've thought of something that actually worked, that if there was an induced feedback loop, we might see its effects during the recovery period from an activating signal. And so what we've done is to treat the wild type and mutant fish with an activating agent and then measure the p53 level at time points after removal of the agent. So if you imagine that the feedback loop is an induced loop, then in the case of the wild type, you should turn on the feedback loop and you should see the clearance of p53 rapidly. Whereas in the case of the mutant, because it can't turn on the MDM2, then what will happen is the question. And the theory is very straightforward. So we imagine that in the absence of stress, MDM2 is able to control the level of both mutant and wild type p53. When we have a stress signal, this ability of MDM2 is blocked, say by phosphorylation at that ATM site. It can't interact anymore, and p53 accumulates. But then when we take the stress away, in the case of wild type, we've accumulated lots of MDM2, so there's a very rapid return to low levels of p53, whereas in the case of the mutant, it's high level. And we've done these experiments in tissue culture using nutlin, and it's quite amazing. When you take away the MDM2 inhibitor in a wild type cell, p53 levels start to drop within 5 minutes of removal of the drug. So this system really is very rapidly able to respond. And what we see in the fish is very dramatic. So you can see here treating with roscovitin and then removing the drug. And you see here's the p53 that's accumulated, and it just disappears very rapidly. Whereas in the mutant, it persists. So this fitted very nicely with what we expected, and we were very excited. But when we started to use other agents, things got more complicated. And so here's using camptothecin. And again, the same basic picture. p53 accumulates and then disappears in the wild type. But the degree and level of persistence in the mutant fish was quite remarkable. So long after the drug has been removed, we're still accumulating these very high levels of p53. And with ionizing radiation, it's even more dramatic. I mean, I always worried about drug metabolism, drug half-life, drug persistence. So going over to ionizing radiation was an attempt to make the picture simpler. And what you see here, so now we're irradiating at t equals 0. So we see the maximum accumulation of p53 3 hours after the irradiation, and then it disappears. But look what's happening in these mutant fish. Now, this is just a single amino acid change in p53, a huge change in the level of p53. And you can see it very dramatically in terms of dose as well. So here we're using different doses of ionizing radiation, looking at the wild type, and then looking at the mutant. And you can just see, well, you can see quite a lot here. First of all, the mutant is a kind of hypersensitized background, and this is potentially very interesting for drug screening. So this sensitivity here is very dramatic, and the response is very persistent. But if we're at 10 gray, eventually the system seems to be able to get back to lower levels of p53, whereas at 20 and 40 gray, it can't. But these fish are still alive. So there's a lot of strange things happening here. And we've recently got this to work in a whole mount. This is work from Ho Peng, so just showing the same thing, essentially, just staining the tail. And you can see after camptothecin or radiation, this persistence, which is completely absent in the wild-type fish. So you've got a sensitization and a persistence as a result of this mutation. Now, this is the last thing I want to talk about, really, is why zebrafish is so important for studying this problem, because we would love to be able to study this problem in a tissue culture model. But if you take cells from a mouse that's able to make mutant, either over wild type or mutant over null here, which is the more relevant, or mutant over mutant, you just can't do it. As soon as you put the cells from the mouse embryo, which doesn't stain for p53 at all, put them into tissue culture, they immediately stain. So whatever signal this is easily turned on by tissue culture, doesn't stop cell growth, and essentially cells can exist, if you like, in this kind of stressed state quite happily, and most tumor cells must be in this state. And you can see it very clearly here. So this is what it looks like if you take cells from a mouse that has a single wild-type allele. You get the odd positive nucleus in the culture. But look what happens. You take the same just from this mouse here. And if you treat with nutlin, you can see in these cells this dramatic accumulation of p53. So clearly the maintenance of this low level of p53 in the wild type is totally MDM2 dependent. So the big challenge for us is to understand, exactly what this signal is that's so commonly activated in tumors all the way from fish to man. What is the stability signal, and how does it relate to what we're seeing in tissue culture? So there's lots of potential possibilities for what this signal might be. It could be constitutive ribosomal stress, constitutive DNA damage. Is it oncogene activation? What's keeping the level up so high after that single dose of radiation? Liz Patton has shown there's a very clear defect in nucleotide excision repair in a p53-dependent way in the zebrafish model, which is very apparent but much harder to see in the mammalian models. And it may be because in the zebrafish at the time of development we're looking at, there's so much DNA replication going on that repair has to be really working. Every system has to be working flat out. Maybe that's why we can see it so clearly. And this signals a key attribute of human cancer, so we would really like to pin down what it is. So one thing we've looked at is to ask the question, because one model has been that there is a level of constitutive DNA damage in human tumors, and work from Yeri Bartek and others has suggested that this could be a characteristic of cancer development. You go through a phase of continuous DNA damage signaling. And so we've looked at some of these tumors with the antibody to gamma H2AX, and we do in fact see quite clear staining in the tumor, implying a level of constitutive DNA damage signaling, though it's not in every cell and it's not completely coordinate with what we see in terms of p53 staining. So it's suggestive, but it's not absolutely clear that that's the explanation. Certainly when we look by Western blotting at what's happening in this persistent p53 accumulation after ionizing radiation, the accumulation of gamma H2, the persistence of gamma H2X is very clear. So there is some link here between these two events. So I'm going to finish there on time, hey, and thank everybody who's helped, particularly Guolin and Hopeng, but also other people in the lab, especially Amanda, who helped with the mice, Ismail, who's done experiments looking at the effect of oxygen tension in the system, and Declan, who's helped us very much with the staining experiments. Thank you.
That's great. We have time for questions. Liz.
I can hear. Yeah, I can answer. I'll rephrase her question in a way that I find easy to answer. Yeah. So yes, I should have been much clearer. So roscovitin is a CDK inhibitor. It probably activates p53 through an effect on transcription because it inhibits CDK7, CDK9. But it's not a genotoxic agent. It's gone into clinical trial and had to do a lot of work to show that it wasn't genotoxic. So at least that's one potential difference. So in one case, we are delivering a non-genotoxic signal that's stabilizing p53. So I can explain the roscovitin result in a sense on that simple model. So it just takes, there's not much MDM2. You've accumulated a lot of p53. That MDM2 has to kind of grind through getting rid of all of that p53, so it takes time. But I think what must be happening with the radiation at the higher doses is that somehow the system's getting locked into a continuous feedback, feed-forward loop in a way. So one possibility is that mutant p53 itself is kind of DNA damaging. So, if you imagine that p53 is involved in nucleotide excision repair, and then you overproduce a protein that's kind of a dominant negative, you can kind of imagine a scenario where you're continually creating that situation. And we have some evidence in the mouse model that makes us think that way. I mean, it's very provocative, but we think the dose of p53, the amount of p53 is very, very important in flipping over to that locked-in state. But that's, this is hand-waving, but it's good for the bar.
Tom.
Yeah. Yeah.
Yeah, I thought so. Now is it working?
Yeah, very powerful.
This is so fascinating, this accumulation in tumor but not wild type. Is that due to this kind of chronic DNA damage going on, or MDM2 inhibition, or how are you?
Well, I think, the simplest model is that it's exactly that. So in tumors, there's a constitutive signal to the DNA damage pathway, and that signal is continually inactivating MDM2. And that's a model. And certainly if we overexpress massive amounts of MDM2 by transfection into a human tumor cell line that's expressing very high levels of mutant p53, we can make that p53 go away. So the p53 is still a substrate, implying that it's the MDM2 that's lost function. And I think that's the most likely explanation. But there may be, we still don't absolutely know. So we need to really look hard at the post-translation modification of MDM2 in human tumors and those kinds of issues to try and really resolve this. But what's interesting to me is it's a very sensitive signal. There is, clearly this is something that's easily triggered, and cells can grow quite happily in this state. So it implies a kind of tolerance of genetic instability in tumors that I think is very important.
I guess it could potentially be druggable, right? The
Yes,
pathway.
indeed. No, absolutely. I mean, if you had a drug that only killed cells that were accumulating mutant p53, you'd be very happy. And I think it's a really good thing to think about.
Along the same lines, have you looked at any lesions before they develop tumors, like in the eyes where they're developing the schwannomas? In other words, is the accumulation of mutant p53 a marker of the pre-malignant state in these tumors as well? And is it heterogeneous, or does it become homogeneous later?
Yes, I mean, we haven't done much in the zebrafish, but there's a lot being done in the human system that you can kind of model, and it does seem to be dependent a bit on tumor type. So in skin and in the fallopian tube, it looks very much as if this accumulation of p53 is a very early event. The event's much more frequent than the tumor occurrence, and you see it in cells that are morphologically almost normal in appearance. If you could microdissect and sequence, it's mutant p53, so the mutations happen very early. If you look, for example, in colon, you don't see that at all. So in colon, APC mutation happens long before p53 mutation, and you have a long period of adenoma, and then you get the p53. So it may be to do with the mutational burden or particular circumstances. And obviously skin UV, people have argued in the fallopian tube that aspects of steroid metabolism may give rise to a DNA damaging or mutational environment. But so you really are asking sort of questions about how early is point mutation in my cascade, and I think it's a really interesting question. So it would be great to try and get at those questions in the fish model as well.
Over there.
Very interesting. So going back to the drugs and the different p53 accumulations, one of the differences between the drug treatments and the radiation treatment is the time of the cell cycle that they may act.
Yes.
So did you look in tissues whether there was accumulation in different cell types, because they could be in different parts of the cell cycle?
Yeah, I mean, I think this is a really good suggestion, and we've been struggling a bit because of my incompetence and various other difficulties to try and get the cell cycle marker and the p53 marker going exactly in a way that we can really pull this apart. And I think it's particularly important because we really need to look at exactly at cell cycle stage. And in the case of the wild type, of course, the p53 will block the cell cycle. So it's very difficult to untangle. But the great thing about the mutant situation is, because we don't think the mutant is causing any cell cycle arrest, we can look at the accumulation of mutant protein in correlation to cell cycle very clearly. I mean, from talking to Joan Heath, the patterns we see in our staining look very like a BRDU pattern. I mean, there's a clear relationship between cells in the S-phase compartment and the cells that stain, if you look at the particular developmental stage that we're looking at. And obviously, we'd like to do that in great detail.
Over here. Okay, go back. That's good. Great. Now I don't have to run.
So you're mainly studying the accumulation of mutant p53, but how important is it to check the low level? I mean, before it actually accumulates the mutant p53, because also if you look at the cell types, the cell lines that you just showed, you can already see like the multiple nuclei in one cell.
Yes.
Yeah.
Yeah. No, I mean, it's all a question of threshold. So, I mean, as our antibodies get better and our techniques get better, I think we can, set the threshold. So we're just beginning to see what I would consider to be the normal level, just detectable. And it's, we have to think about going to more sophisticated staining methods, I think, because we need to be much more quantitative. I mean, the peroxidase looks great and it's very clear, but it's not very linear. And I think we're going to have to, go to a fluorescence-based method and start counting things to really get at these issues. It's a very dramatic change in level, as you can see from the immunoblots. But we need to look in more detail. If this, because we know there's haploinsufficiency, we know that the exact level is very critical for these pathways. So anything in p53, you have to be able to deal with haploinsufficiency.
Yes, over here. Oh, sorry. Here, use this.
Thank you. Thank you very much for your wonderful speech, Mr. Lim. My question is, why does p53 mutation specifically cause tumors in eye, growth plates of optic teflon, liver, and intestine? What do you think about the mechanism? Thank you.
It's an easy question.
That's dead easy. I don't know. I mean, I mean, we can't answer that question. I mean,
Oh,
obviously,
It's
p53
good we have one question
Yeah.
you can't answer.
Yeah,
It's
exactly.
good.
Yeah.
Can you comment on in the fallopian tube slide? It seemed like there were cells that looked atypical that didn't express p53. I'm wondering if you could comment.
Yes, I mean, it's very interesting. So one of the possibilities, of course, is what we would call the silent lesion. So you can imagine a situation where you stop expressing p53 completely, and you could get the same phenotype, and those wouldn't be spotted. And in our examination of human tumors now that we've got the threshold of staining so that we can actually detect the very low level of p53 in tumors that are wild type, we definitely can see tumors that are null. And the null phenotype does occur. So if you think about what's happening there, if you think about the skin and Brash's work, there have to be invisible lesions, right? There have to be, at some frequency, there will be point mutations that are stops, and you just wouldn't see it, and because you can't see an absence of nothing. Right? So it does mean that the burden actually is even higher. And if in the clonal sequencing studies now in serious ovarian cancer, where the mutation rate in p53 is about 97%, about 30% of those are stops. So I think the frequency of stop mutations in p53 is generally underappreciated, because when you see lots of nice mutant protein, you're happy, but there's clearly another group there.
What do you think in the ribosomal protein mutations? They lose p53 at the protein level.
Yeah.
So, but this is even with an allele where there's mutant proteins. So
Yes.
do you think there's a drive in those ribosomal mutants to really become null?
I mean, that's an amazing result, and I actually was, it troubles me a great deal, and I'm trying to think. I mean, it's very striking data. It doesn't happen in the normal tissue. It's a tumor-specific event. It's associated very clearly with those mutations, and it hasn't really been, the human equivalent of that piece of biology hasn't yet been picked up. But it's so striking that one would imagine it would be. I mean, the only thing I've done recently that kind of made me think again was we were just looking again at the E6-driven degradation pathway, and I realized that it's so effective that you can get the appearance of no protein, lots of message, everything fine, and it's just an incredibly active E3 ligase. So I just, it would be worth sort of thinking about experiments to try and get at that issue. And the other thing that's weird is I used to think proteasome inhibitor deals with that problem, but no, there's quite a lot of these proteins, these E3 ligases that have a dual activity. They actually take the protein to the proteasome and feed it to the proteasome. They kind of, the ubiquitin system's there to aid and abet, but it's not the only part, and so there's still potential things there.
Great. Okay, we'd like to move on to our next speaker, who's Jim Amatruda. He will change the title of his talk to Genetic and Genomic Approaches, Understanding Germ Cell Tumor Pathogenesis.
Thank you, Len, and thanks, of course, to the organizers for this great opportunity, and to our hosts here at Cold Spring Harbor, Asia, for this spectacular setting for a meeting. And I wanted to share with you a story from my lab that's been going on for a few years, and that's really pushed us to use the developmental and genetic aspects of the fish to understand something about human tumor biology. And in particular, we're trying to integrate genomic and genetic approaches. So maybe a less pretentious title for my talk would be Fish and Chips. Okay. So the area we're most interested in is germ cell tumors, and these are tumors of the germ cells that will eventually form the testis or the ovary, so the forerunners of the germ line. And they're a relative minority of all childhood tumors considered as a block, but in the adolescents and young adults, they actually become quite an important group. And it's the most common cancer in young men is testicular cancer. And these are in some ways a paradigm of a very curable cancer. So to an oncologist who treats adults, this is one of the easy ones, because even patients with quite advanced stage disease can be cured since the advent of cisplatin in the 1970s. But there's a number of areas where we still really need to make much better progress. For one thing, there's about 15% of patients are not cured, and in particular, there are patients with troublesome clinical categories, those patients with sort of what's called poor risk disease, bulky tumors, high tumor markers, or when tumors occur outside of the gonad, these patients do much less well. And in fact, when you get combinations of extragonadal tumors that are resistant to chemotherapy, essentially all those patients will die of their disease. And one reason is that we really only have one good drug, which is cisplatin. So once that's exhausted, we have very, very few alternatives to offer the patients. And because we don't know very much at all about the molecular pathogenesis of these tumors, we don't have a rational way to apply targeted therapy as an alternative. The other problem we think about quite a lot in kids is that the toxicity of these regimens is quite significant. So toxicity targeting the kidneys, the ears, lung damage, early cardiovascular death. And here's an example in which you can see that the probability of developing significant hearing loss is about 80% in young children treated with cisplatin-containing regimens. So we have quite a lot of incentive to find better approaches through understanding molecular pathogenesis. We know something about the development of germ cells. So normally, primordial germ cells will migrate through the embryo to establish the gonad. And these embryonic germ cells undergo differentiation to either according to male or female fate. In the case of the testis, you can see this structure here with these nice spermatogonial stem cells that undergo programmed differentiation to form all the other cells in this tubule, ultimately culminating in the mature sperm. In the tumors, something goes wrong pretty early on. And I say that because there's evidence that the tumor cells recapitulate many of the features of embryonic germ cells. So in the tumors of adolescents and adults, there's a precursor here known as carcinoma in situ or intratubular germ cell neoplasia. So you can see these neoplastic cells accumulating. We actually don't know their p53 status. It'd be very interesting to know. But because these are tumors of pluripotent cells, they can really take on almost any kind of developmental fate and have very different histologic appearances. So there's a group of tumors called seminomas or germinomas, and these are really just undifferentiated primitive germ cells. So these look and have the gene expression profile of early embryonic germ cells. On the other hand, the tumor cells can differentiate all the way to a teratoma, which is a tumor representing all three different germ layers, or even extra embryonic structures like yolk sac tumor. And these are fascinating developmental questions, but actually also very topical clinically, because these tumors of different histologic differentiation types behave differently in the clinic. And we really need to understand better how these developmental decisions are made to try to understand that impact. So we got interested in this a few years ago when we found a fish, when I was a postdoc in Lanzon's lab, in which the males developed these large gonadal tumors, which turned out to be testicular germ cell tumors. And if you look at the phenotype of these tumors, it's quite similar to the human seminoma. So here's the normal zebrafish testis. Here are the spermatogonia, which will undergo this orderly differentiation process. However, that differentiation is completely abrogated in the mutants. This was an ENU allele that arose during a forward genetic screen. And so differentiation is really blocked here, and these cells express the markers of early germ cells. So we assembled good old-fashioned pedigrees. Unfortunately, the mutation was quite penetrant, so we were able to collect animals that develop tumors and do positional cloning. And Joni Newman in my lab ultimately arrived at the mutant locus, which turned out to be a mutation in this gene called ALK6B, also known as BMP receptor type 1B. So this is a point mutation that introduces an early stop and removes this kinase domain from this transmembrane serine-threonine kinase. So a little bit of nomenclature here and sort of fish-specific biology, because one of the initial puzzles when we cloned the gene was that these fish did not have a striking developmental phenotype. And as we've heard from Phil Ingham and other talks yesterday, BMP plays a critical role in early development, axis formation, limb development. In fact, the mouse knockouts for this gene have both limb and gonad defects. But the fish, it turns out there's two copies of the gene, two paralogs, one of which sort of takes care of the axis formation. And this form, which is relatively gonad specific. We think that's why really the only lesion we see in these fish is gonadal tumors. This receptor is part of a heterotetrameric membrane complex that binds to BMP ligands and transmits a signal through phosphorylation of intracellular SMAD1, 5, and 8. And this phosphorylated SMAD binds with the co-SMAD to enter the nucleus and transactivate BMP-regulated genes. So there's two sort of convenient readouts for this pathway, phosphorylation of SMAD and BMP-specific gene regulation. And we could see that, as predicted, in these fish, both those pathways are disturbed. So this is phosphosmad staining with a phosphosmad-specific antibody in the normal testis. And you can see that the staining comes on in a few cells and then goes away. And this actually, I think, will be important in a minute. In the mutants, in contrast, there's really no BMP signal transduction activity in the testis tumors. In addition, when we look at expression of canonical BMP-regulated genes like ID1 and ID2, they are defective in the tumors, and a BMP-repressed gene called midkind is overexpressed. So with this, and also with transgenic rescue with a wild-type allele, we were confident we had arrived at the mutation explaining this phenotype. We really wanted to know immediately, does this tell us anything about human cancers? So we know that in the fish tumors, BMP is required at least transiently for differentiation of the tissue. This fits with what's known from mouse spermatogenesis, where again there's transient BMP signaling in the spermatogonia as they initiate differentiation. So the question was, does this pathway contribute to the differentiation decisions made in human tumors? And so we assembled clinical specimens of these different tumor types and stained them again with the phosphosmad antibody, and found that indeed the seminomas, which are the undifferentiated tumors that resemble the fish tumors, are really have absent BMP signaling activity. And interestingly, there were some tumor types like the yolk sac tumors, these non-seminomas, where the BMP pathway was very strongly on, as it was in this precursor cell and in the normal testis. So this forced us, I think, the value of this is that it forced us to think about these tumors in a very developmental context. So much of the excitement and clinical therapeutics development in cancer these days is focused on cell proliferation, DNA damage, cell death pathways. But especially childhood tumors appear to be often driven by developmental pathways, Wnt, Notch, Hedgehog, TGF-beta, BMP. So we really need to understand the role that these pathways play in human tumors and to have animal models in which to sort of extract the biology. So the reason I think this is important is that there's a clear relationship between the developmental state of the tumors and the sensitivity of the tumors to chemotherapy and radiation. So seminomas, these undifferentiated tumors, are really notorious for being exquisitely sensitive to chemo and radiation. At the other extreme, a teratoma really is almost by definition a chemo-insensitive tumor. These are differentiated and not dividing very fast. It's more of a surgical problem. And in between, there's this class of non-seminomas. So these tumors are somewhat differentiated, and it turns out that they are, in general, more resistant to chemotherapy. And even within a given tumor class of a type of seminoma, the presence of, whoa, I don't know what I just did. The presence of these differentiation-promoting pathways and expression of genes such as BMP7 turns out to correlate with the resistance of that tumor to therapy. So it's as though there's sort of a gradient or a continuum of BMP signaling here, whereas if it's completely off at either end, the tumors could be, these tumors are very sensitive. These tumors are sort of done developing, but these tumors are locked almost into a state with persistent, abnormally persistent signaling that makes them more vulnerable or more resistant to treatment. So to explore that idea further, we also considered this very interesting finding that this carcinoma in situ stage is BMP signaling is on. It's as though this developmental pathway has never shut off. And then there must be other mechanisms that we're currently exploring that take you from this state down one of these different pathways. So in order to understand that, we clearly need to know a lot more about pathways other than BMP. And so part of what we're doing in my lab and through some collaborations is to take a broad view of human tumors. So that was the fish part. This is the chips part. So we're doing several different approaches, including expression profiling in collaboration with a group at Cambridge in the UK. And we've done sort of standard arrays and are now doing RNA-seq to look for non-coding RNAs. And initially what we've seen is that some genes, including BMP2 and BMP7, driving this pathway are highly expressed in yolk sac tumors. Similarly, we did in collaboration with Jen Pointer at the University of Minnesota, methylation profiling. And in there we found that there was a group of co-regulated genes in yolk sac tumors, these non-seminomas, in which, although the tumors overall are highly methylated, there's a group of specific group of genes that are hypomethylated and more highly expressed, including many Wnt pathway targets. In my lab, we're doing now copy number variation analysis of human tumors, in which we found that there's specific copy numbers associated with different disease types. Overall, the goal here is to, of course, to look at the copy number variations that might correspond to resistant or sensitive disease. But even at the beginning, we've started to see amplifications of some of these genes in the BMP and the Wnt pathway, pointing us to, again, the importance of these developmental pathways. Finally, now we're doing exome sequencing of matched tumor and germline tissue, and this will hopefully give us not only pathogenic SNVs that could be targets for therapy, but further insight into other mechanisms by which these genes are, these tumors are driven. And I think the message from this overall is that we're going to get a list of aberrations, genes, or mutations, and we're going to need these in vivo models in order to work out the biology. I think that the fish will serve for many, many years as a powerful model for this kind of approach. So the findings are exciting and intriguing to us, but I think it's fair to say that neither BMP nor Wnt is currently a very well-targeted pathways for human cancer, but it made us think about what might be downstream of these tumors, and particularly the mTOR pathway. So as we'll hear from Jeroen and we'll hear from others in this meeting, the PI3 kinase pathway downstream of receptor tyrosine kinase is important in a number of pathways, one of which is the mTOR complex, which has, at least mTOR complex 1 has targets including ribosomal protein S6K and 4-ABP that help drive translation in tumor cells and a growth phenotype. And we found that treatment of human germ cell tumor cells with either BMP2 or a Wnt conditioned medium potently activates this pathway. We have some evidence that Wnt actually is also transactivating BMP at the same time. So the presence of active Wnt and BMP signaling in non-seminomas appears to be converging on the mTOR pathway. And we have some insight now into the mechanisms that I won't really go into in the interest of time. But the exciting point here for me is that this is a very druggable pathway. So we checked immediately in human tumor specimens and could see indeed that, as predicted, the mTOR pathway is strongly activated in non-seminomas compared to seminomas. So this is immunohistochemistry for phosphorylated mTOR or phospho-S6, both activators of indicators of active mTOR pathway activity, as well as the downstream targets cyclin D1 and HIF1 alpha. And from a translational perspective, these make very nice targets. So rapamycin and other rapalogs are already approved for use in children. And there are other agents now clinically in development, such as BZ235, which is a dual PI3 kinase mTOR inhibitor. And we can show that these drugs impair the viability of these cells. And even at low doses, which don't necessarily kill the cells, but shut off the pathway activity, we can show very nicely that these agents inhibit the growth of the cells. So we're going to, based on these results, we're opening a clinical trial for the use of mTOR inhibitors in children with relapsed or refractory germ cell tumors. And I think building on these results will give us a lot more leverage to apply rational targeted therapy. I'd just like to say that, instead of turning this off, that we still have a lot to learn from the fish. So we've now shown that adding into our initial model other lesions in the BMP pathway lead to increase in tumors, either BMP ligands or the SMADs, as well as defects in oogenesis, which we've seen in our model that I won't talk about today. And that the Wnt pathway is proving also quite interesting. So a student in my lab, Clayton Bolt, has now shown that treatment of developing embryos with small molecule Wnt inhibitors during the periods of critical germ cell migration strongly impairs the migration of the cells. And this may have some relevance to the development of extragonadal germ cell tumors in children. So I'm going to skip the summary slide since I'm over time, but I thank you very much for your attention. I'd be happy to take any questions, and I want to acknowledge many of the people who contributed to this work, including Kenneth Chen and Abe Shukla, who did the human germ cell tumor studies, Joni Newman, who, I wonder what this says, right? Joni Newman, who cloned the gene. Vanessa Demoulis, a student in my lab, has this is the danger with Google Translate, isn't it? Has a poster that I hope you'll all go see today, and I'd like to thank my collaborators as well. Thank you.
All right, somebody has to tell us what it said.
It should be 谢谢. It's not, I think Jim, you translate acknowledgement, right?
We'll
Should be
we'll talk about it at the bar. Okay.
Okay.
You have a question, huh?
Jim, I want to know that this sequencing result, I know the time limit, because there is a report that the normal testis development and the cancer occurrence share some genes and the pathways. So I thought you list several sequence projects to do this analysis of tumors and the testis of this the seminoma. I wonder you have find some, like they reported, there is some genes shared by the two process. Have you looked at them?
Yeah, so I think the question is the sort of convergence of normal developmental signaling pathways in testicular or germline development and tumorigenesis. And I think we just have early days on the sequencing results, so we're seeing some intriguing things. In each case, we're matching, of course, the tumor with the normal germline. One of the shocking things is how many germline mutations we all walk around in and apparently important genes. So I think I'll have to wait till the next meeting to report more on those results.
Okay, thank you.
Dr. Look.
Jim, I wondered, in the yolk sac tumors, have you looked for PTEN mutations? That would seem like a likely...
We have looked a little bit. Others have looked, have not seen them. We've seen some actually APC mutations, but so far not PTEN. But it's early days yet.
So it activates mTOR from upstream rather than the usual PTEN mutation.
Seems to be. So there's evidence I didn't show that the MAP kinase pathway is also quite active in those tumors, and we think there's crosstalk. But some of the BMP and Wnt may sort of bypass AKT, we think. That may be the mechanism.
Troy?
Very nice talk. What's interesting is that clinically, by and large, these children show up inevitably with distant metastases upon presentation. And I'm wondering if some of that can be explained by the SMAD dysregulation that you see.
So it's an interesting question about patients presenting with advanced disease. So I think one of the things we're specifically interested in is comparing localized to advanced disease. And I think we don't know yet, but whether some of these migratory pathways are playing a role, it'd be interesting to see.
One last question, Dr. Shardle.
Jim, I think your BMP receptor mutant is a null or a knockout mutation. So you would expect that taking away the BMP would make the same tumor, right? But then you show that in the human tumors you have amplification of BMPs. So how do you explain that?
So I think the question was the fish model is a loss of function. We see loss of BMP signal transduction activity, and when we add in haploinsufficiency for other BMPs, we see an increased tumor incidence. And I think this is most like the undifferentiated seminoma type tumors in humans. The finding that this pathway sort of remains abnormally on in the non-seminomas, that may be its own sort of aberrancy. And so there I think it's the gain of function that's sort of dangerous. And this is something we have to do a lot more work in. But I think the fish is a model of one type of tumor but not the other.
Okay, I think we're going to move on. Thank you, Jim. Wonderful talk. Our next speaker will be Keith Chang, who's going to talk about high-throughput whole animal 3D imaging at cell resolution as a basis for computational phenomics.
Hi. Thanks so much for the organizers. I thought I'd pull up a fisherman since we're fishing for answers. And if you look closely at that picture, there actually appears to be zebrafish in there too. So the main points I'll try to go through today is that if we must achieve cell resolution, and that resolution historically in the history of science and medicine is what's responsible for us for entering the modern world of what we're doing today. Studying the whole animal is important. I think we've seen a number of examples of phenotypes that cross from one's target organ into others. And we've demonstrated that semi-quantitative scoring of phenotypes actually allows one to cluster genes by function. For chemical phenomics, well, there's a lot of drug screening being described here. And in addition to looking at the full range of effects across tissues, we need to also deal with the issue of chemical toxicity in the environment, which of course includes toxicity to our unborn children. The number of data points that we're hoping to be able to extract from all this in the future, it's going to require high throughput. There's no way on earth, even if you collected all the current world's pathologists, that you could actually do scoring quickly and reproducibly. So there's going to be a lot of speed enhancements that I'll describe a little bit about with time, if we have enough time. And finally, the data that we want to generate, I think will have to be web-based. File sizes are so large that there's no other way to do it. Before I forget, I'm going to describe quickly the groups of people that I've been involved with. This is a highly interdisciplinary project. I have people on my side and in other institutions who are physicists and engineers, and we have histology, as most of you know, and I'm talking with toxicologists, and there's so many people in the community who have helped me, I can't even mention them all, but there's a lot of computing, and we recently formed a graphics arts team. You'll see why in a minute. So I thought I'd point out for fun that this was a statement from before DNA structure was known. Test tube of cancer research is not just of glass. It is more often in the living and genetically controlled experimental animal. This is 1938. So the Phenome Project is something that the zebrafish community is beginning to pursue. By making mutants for each gene, we hope, we're going to be able to probe the function of every gene in the genome, in this vertebrate genome. And other labs concentrate on the physiology, behavior, and I'm concentrating on trying to solve the issues that morphology brings up. I don't know that I have to convince any of you of this, but imaging and Jim just spoke to some of this, is the basis for diagnosis, and in radiological imaging, all the way, and lower power imaging has been used for genetic screens of ours and others. It's just a centrally important thing, and I have to show this slide. It's from my pathology residency, where I was inspired at this time to study cancer from the standpoint of why are cancer cells looking like this, and what is the cause of genomic instability. I don't have time to talk about either of those, but except the peripheral thing. Now, so I'm in China, and so I'll mention again that we named the first, I think it was the first zebrafish mutant that's Chinese. It's called Hulihutu, which means confused. And you can see why these layers, which are beautifully layered, and just like the human, are confused in this mutant, because I was asking, what are the genes involved in causation of some of the Z-typical nuclei that you see in human cancer, and this is a mutant. So this is a clue, and it turns out to be a polymerase subunit, as you'll see in a minute. For the moment, I thought I'd point out perhaps what's obvious, but for the students perhaps more. We talk about biological function. These are things that we know are basic functions of our body, from pigment to neurological and et cetera. And that's something we should keep in mind for the genes that we study. We need to also study the biochemical mechanisms, of course, and know what cells are being affected in any given mutant. And even though we're quite justified in focusing on one organ system at a time in our work, that's the only way we can get funded or published. It is important in a phenome project to go the opposite way and to be able to look more comprehensively at all the genes' functions, which is our purpose in this case. So what's a phenome? Phenotypes are what we get when you treat when you have a mutant or when you treat with a drug, and the whole gamut of possible phenotypes is the phenome. It's an imaginary construct, however, because we'll never think of all the possible screens. But to approximate a phenome, you collect information that you can with whatever assays you do have. Now, for any given gene that you might mutate, there's no way to predict which organ's affected or when that organ or organs will be affected, which means that you have to look at the whole animal, each tissue, and that you have to look at multiple developmental stages. This is a big challenge for the Phenome Project. So what are we going to do about this problem? Okay, so in a recent paper we published that reviews different types of imaging that you can do. These are the types of tools that you're all familiar with. MicroCT perhaps a little bit less. But if you ask yourself, and MRI is one of them, but typically many of these have resolution that doesn't quite reach cell resolution. I thought I just got these data a few days ago. This is a 20 Tesla MRI that we've done with some Zebrafish just for, to see how useful it might be. It doesn't have cellular resolution. We got down to 8-micron voxels, but we are able to segment out organs, and if there is a computational way to segment each of these out and automatically measure things, it might become a useful tool. And these are the kinds of contrasts that we were able to generate using this tool, which is very slow. So which of those tools give you cell resolution? So you have 4 things, and confocal microscopy is fantastic for living fish, but it has a very small field of view, unfortunately, so you can't see everything at once. And also you can only see whatever is fluorescent and not what is not fluorescent, obviously. Histology allows you to see everything, and it has a great field of view, but it's destructive. So once you slice the thing, it's all over. Blockface, it's so slow that you wouldn't want to consider that for any kind of screen. And MicroCT is where we think we'll be going. I've already told you how fluorescence confocal is so important, and that you can use blockface, but MicroCT is really the only way I hope to show you on which you can do whole animal cell resolution imaging that has potential for high throughput. So we did that genetic screen and found Hulihu2. This is already published work. And we accomplished that using a mold. In this case, it was a 32-hole mold into which we stuck 2 larvae per well. We currently just ordered a set of molds that's of the next generation type, and we'll hopefully be giving those away at the Zebrafish meeting, or you can write to me, and we'll try to do that. So Hulihu2, we positionally cloned, and the region containing the gene, this is the old-fashioned way. We had 5 different genes, and 2 of the knockdowns down on the bottom looked like the mutant top right. So to define which of the mutants, which of the knockdowns is the gene of interest, we used histology for our positional cloning and found that the lower 2, the one on the right seems a little bit more like Hulihu2, and it turns out that's the gene by other pieces of evidence. And very interestingly, at the Asilomar conference many years ago, I walked by Ishiro Masai's poster, and there was a picture of the left side of the lower fish, which is a mutant of his, and I said, "Did you look at the gut?" He said, "No." So we looked at it, and it turns out that it actually looks like Hulihu2, and it turns out to be another subunit. These are 2 subunits of DNA polymerase alpha. Okay, so you cannot really, in human diagnosis, really understand what the disease process is without seeing cells. This is the mold that we're going to be trying to give away, so, and that we use to develop the tools that you see that we have on our website, Zebrafish Atlas. So we asked, I'll go over this pretty quickly, this graduate student, Georgia Thomas, pursued the question of what could we learn if we looked at the mutants that Nancy Hopkins generated by histology. If you see additional resolution, what can we get out of that? So we looked at about 100 of those, and we scored a lot of things over 2 years. And we to summarize those years of work, you can look at how many of those 97 had one organ affected, and it's only 11, which tells you that you really need to look beyond the organ of interest to get a complete idea of gene's function. On the bottom left is was online about this foie gras mutant, and on the right side is some of the information that we accumulated regarding this one mutant. So there's a lot more depth of information across multiple developmental stages, in fact. We looked at days 2, 3, 4, and 5, and looked at the increase, and this is something one could potentially do. It might be a little bit too involved for the Phenome project, but it's the kind of thing one could do. We did it by human inspection, but if this can be computationally done, it would be, of course, much faster, and then it would be possible to do this for the Phenome project. So it turns out that if you look at the sets of changes, the phenotypic signature of each mutant, and then you asked which ones were similar, this one group of actually protein folding complex subunits had a similar signatures, and this demonstrates the possibility that cluster analysis based on phenotype could be valuable. And of course, we have genes such as SOX9 that have very specific effects on cartilage. This is a single tissue effector, at least in these days that we looked, and that is reflected by what we found. And perhaps most strikingly, this work was also done with Brian Canada, top right there, who's a bioinformatics person. And you can see that under integument and under kidney, for example, if you compare what you can see with a stereo microscope versus histology, it's quite strikingly different. Now, one thing I thought I'd mention here, and I'm going to have to move quickly now because we're going to want you to show you some pretty pictures before I leave. So if you actually grade the phenotypes, you get more information than if you don't grade them, which is on the left. You basically separate genes in terms of their signature. Okay, so I think I'm going to move on here. It turns out that Zebrafish, of all of the vertebrate model systems, is the only one that's small enough to get cell resolution if you divide the anatomy across the space of a chip. This is Darren, who did a lot of this work with me, and this is MicroCT, whose basis is like our CT, except 1,000-fold smaller units. And you rotate the specimen in this case. We'd get dizzy, but you rotate the specimen and take 1,500 projections and calculate the 3D volume. So it looks like this doesn't want to work. Hello? So the source of X-rays is on the right side, and you move the fish to different segments to get the whole fish. This happens to be a tabletop unit, and confocal is just parallel beam. The synchrotron is parallel beam. So this illustrates the 3 steps. We collect projections. We generate slices, of which one is shown in the middle, and then you can look at 3D renderings. And I'll give some examples now of what we've been able to do. Looks like we have to go pretty quickly. This is comparisons. I'm going to show you a bunch of comparisons between histology and MicroCT. You can see detail on the left, not so much on the right side. The gills are on the top right there, and this is the same 33 gigabyte file yields each of the other 2, just cut different directions. This is a lower of the 2 resolutions that we can do. This is the eye, and those dots are individual nuclei. The long dots are red blood cells and top side of capillaries. This is Hulihu cut 2 different ways. These are several comparisons between histology and MicroCT. We have islet in the middle, and various other sections there. The pseudobranch is on the top left. And I'm just going to flash through some comparisons so to show you that we have pretty reasonable detail. We're actually able to tell the different cell types from each other at the higher resolution. All right, so the visualizations we're able to do are important, and I asked whether we could model individual cells, and This is work I showed last year, but this can be modeled by computer. This is not human generated. This is generated by the computer from our MicroCT images of the larvae. You can count nuclei, and there are multiple types of visualizations you can do. This is one of them. I've gone over time. This is, well, I can go over some of these with you one-on-one. You can actually see the cilia of the olfactory epithelium, for example. Okay, those are, we have element-specific imaging we can do, and the last ones I'll show you are these now. This is a rendering using VG Studio Max, and we're able to cut into the fish, and perhaps I should really stop. You guys want to see this? There's only a couple more movies left. I'm going to have to cut the last part of this one out, and this one's important. You can take the same file now and change the imaging values, how you look at it. You can come different directions and look at the fish from different angles. So that's, I think I'll stop there.
Okay, thank you, Keith. Questions, if we could have the lights. Questions, anybody? There's a question coming up.
So, Keith, the images are exquisite and, yes, cellular resolution, but how are you going to get enough beam time at the synchrotron to do the phenome project?
I'm sorry, could you repeat that last part? How are we going to...
How are you going to get enough beam time at the synchrotron to do the whole genome project, phenomically?
As I had on the one slide, we're going to have a central place that the mutants are sent and samples generated. The good thing is that when you generate the samples, I want to point out, you image them, and when the next method comes out, you can image the same samples again.
But you actually have really good access to beam time is, I guess, the basic question.
Oh, you talk about the synchrotron.
Yeah, so the MicroCT resolution that you showed is all done on parallel X-ray beam MicroCT at the synchrotron, which is a resolution you're alluding that we need.
That's right.
And therefore, all the mutants would have to go through the synchrotron.
That's correct.
And I'm kind of asking whether you've finagled the ability to do that or not.
The current ability is not there, but we have talked with people at Argonne, and they are interested in pursuing this if they get money. So I think that's one of the challenges of this project is funding, and I'm pursuing that as best I can.
Rich?
Do you have a sense yet of the mutants you've looked at by these techniques, that what's the lower limit of resolution? In other words, if it's a subtle change in cellular numbers or cellular phenotype, how few differences can you pick up using these techniques in an automated fashion?
Well, one way to answer that question is that the larger samples, the juveniles, for example, sorry. Yeah, so, I got the speaker and microphone. Yeah, so the larger specimens, we have to use a lower resolution, and there it's difficult to see cells. The neuronal structures are difficult to see, although the tracks are evident. You'll not see capillaries, for example, which is really needed for people working in vascular development.
Last question.
Hiya. So you told us about how important it is to disseminate this kind of thing, and with such huge files, can you tell us a little bit about the kind of infrastructure you'd imagine we'd need to
the infrastructure we'd need is supercomputing centers, and we've been talking with the people at University of Chicago, for example, to do this via supercomputer. I think it's all possible.
Okay. So our next speaker is Jürgen de Hertog. He's going to talk about PTEN and zebrafish tumor-genesis and development.
Thanks, Len. I would like to thank the organizers for organizing the meeting over here and for the opportunity to speak at this very nice meeting, ZEM5. I think I've been to all of the other ones, so it's good. So I'm going to talk about PTEN. And usually, when I introduce PTEN, I would say that PTEN is the most important tumor suppressor gene. But now, after David Lane first talk, I probably have to say that it's one of the most frequently mutated and one of the most important. But actually it is. So in reviews, you will find that people usually say that PTEN is one of the most important tumor suppressor genes, second only after p53. So let's settle for that. Anyway, so PTEN is a lipid phosphatase. It has a phosphatase domain and a tenzin homology domain. And then, like I said, so somatic deletion or mutation in humans leads to tumors. And there are also germline patients known with germline mutations, which leads to these syndromes down here. The pathological features of these syndromes are multiple benign tumors and an increased susceptibility to malignant cancers. Now, the mouse knockouts, the homozygous are embryonic lethal early on, day 8.5. Heterozygous mouse knockouts develop tumors, and actually slight reductions in protein levels of PTEN already increase tumor susceptibility. And slight reduction, meaning 20% reduction of PTEN protein level already leads to an enhanced incidence of tumors. Now, zebrafish have two PTEN genes, PTEN A and PTEN B. We obtained through tilling stop mutations well upstream of the catalytic site in both PTEN genes, and these both are inactivating, and there's no functional protein produced in these homozygous mutants. Now, fish can live with a single PTEN gene. The PTEN A - fish are perfectly normal. They're viable, fertile. So are the PTEN B fish. The double mutants, they're embryonic lethal at around day 5, and they show all these hyperplastic and dysplastic changes, enhanced proliferation and survival. Now, this is specific. We can rescue largely this phenotype by injecting PTEN RNA, and so they are largely rescued at 5 days post-fertilization. Now, I'm going to talk about this story from Suma, a postdoc in the lab. So this is a screenshot, or a shot of her desk, rather. So it's clear that she brought her private life into the lab. And there's a reason for that, because she's working very, very hard. And actually, so this is Suma over here, and here's a picture of her as well as here and over there. So the story I'm going to tell is really single-handedly done by Suma. So what she found is that she followed these PTEN fish with a single wild-type allele of PTEN, about 300 of these fish, and found that in the period of about a year, they developed these tumors. And these are very similar, so 26 out of these 30 are of a certain type, very close to the eye, as you can see here. So these eye tumors are developing over here. It's both with when they retain a single wild-type PTEN A allele, as well as with a single PTEN B allele, we get similar tumors. We did pathology on these tumors, and it turns out these have all the characteristics of hemangiosarcoma. So these are looking like blood vessels filled with blood, and at cellular resolution, the pathologist immediately said that these are hemangiosarcomas. Now, this work is published, so I won't go into more detail now. But what we concluded is that the haploinsufficiency of PTEN predisposes to uncontrolled proliferation of endothelial cells, particularly, resulting in this hemangiosarcoma. And because these endothelial cells appear to be the main cell type that is found in these tumors, we decided to look at embryogenesis in this KDR-like GFP line, staining all endothelial cells, as you can see here, and we crossed it onto the PTEN knockout background and looked for defects there. And what we found is that there's severe hyperbranching in the trunk region, also in the head region. I'm showing you the trunk region here. As 3 days post-fertilization, there are many more blood vessels forming here These are actually blood-filled, so they are functional, they appear to be functional vessels in this hyperbranching. And then, so we're continuing to look at this, and we may have solved or found that why it's particularly hyperbranching here. But I'm going to tell you a little bit of a different story, because someone was looking at the first incidents that she saw a defect in this hyperbranching, and so she took these KDR-like GFP fish in a knockout background and looked earlier and earlier, and then she saw this phenotype where you're now looking at the dorsal aorta here and the posterior cardinal vein, and here are intersegmental vessels shooting up, and then she time-lapsed this. What you can see, you have to look at these asterisks over here at first. So you see this cell which is bursting into little pieces, and now next this cell over here will be going and does the same thing. So what you have been looking at is was first described by the Herbomel lab in Paris and by Dave Traver in San Diego at the same time. And what this is it's called endothelial to hematopoietic transition, or EHT. And what happens in the wild type here, and now you have to look at this cell, it's bending and bending for a couple of hours, and then at some point this cell is budding off into the PCV, and it moves off to the CHT to form blood. So apparently in the PTEN knockout, this process of EHT is impaired. I'll show you this movie again, so that you can have another look at this, close to this asterisk here. So this cell is budding off, but then blowing into pieces and going, and it dies basically. And then next, this cell goes and does the same thing. So apparently these cells die and go into apoptosis. And that's actually what we saw as well. So we did an acridine orange staining, and here in the double mutant, you see these single positive cells at the floor of the dorsal aorta. That's where they are localized, and you should remember that these are PTEN knockouts. So in these knockouts, survival is enhanced. There's not a lot of apoptosis going on, and we only see these specific cells at this position going into apoptosis in this PTEN mutant. Now with activated caspase-3 staining, we showed that these are indeed apoptotic cells. Again, here's the dorsal aorta, and here is a close-up of these individual cells going into apoptosis at this position. We don't see this going on in siblings or wild-type fish. So these hematopoietic stem cells appear normal up to EHT. So at earlier stages with classical markers, RUNX1 and CMIP, there's no difference between siblings and the PTEN-A double mutants. So there are some differences. You might spot some differences between these individuals, but we've looked at many, many individual fish, and overall there's no difference between the siblings and the PTEN knockouts in RUNX1 and CMIP staining. So the hematopoietic stem cells at the floor of the dorsal aorta are forming, they're there, but then when they go into EHT, they bud off, then they go into apoptosis and die. So what are the consequences of these, the loss of these hematopoietic stem cells? Well, as you probably know, these hematopoietic stem cells give rise to all of the hematopoietic lineages. And they are at different stages. They are located at different positions, and so it becomes a bit complicated here. So in the caudal hematopoietic tissue, there's this intermediate hematopoietic site. This is where the hematopoietic stem cells move to first, and then there's the definitive wave taking place at the thymus and the kidney. The kidney forms only after the PTEN mutants have died, so that's not of our concern. I will be showing you some data about the thymus and about the CHT. The primitive wave takes place earlier and does not involve HSCs that have undergone this EHT. And it turns out that the primitive wave appears not to be affected. So erythrocytes at early stages are normal, they're there. There's blood circulation in the mutant that starts around 25 to 26 hours post-fertilization. That's all normal. And these erythrocytes, however, the number of erythrocytes is declining with time. As you can see here, at 4 days post-fertilization, there are less GATA1 positive cells in the double mutant compared to siblings. But initially, the primitive wave appears not to be affected. And so if we look at the caudal hematopoietic tissue, at 4 days post-fertilization, it's now on a CD41 GFP background, where the low GFP positive cells are actually the hematopoietic cells or the myeloid progenitor cells, and the high GFP expressing cells are the thrombocytes. Then in siblings, you can see both populations occurring in the CHT. In the PTEN double mutant, there are more low GFP positive cells and less high GFP positive cells. We actually counted these, and a quantitation of that is seen here, where indeed low GFP positive cells, so these are the progenitor cells or the hematopoietic cells. In the mutant, there are more of these cells than in the siblings, and there are less thrombocytes, so less fully differentiated thrombocytes in the CHT. Now it turns out that there's enhanced proliferation in the CHT, so this is a phosphohistone 3 staining of the sibling. You see the odd cell being positive here, and in the close-up you can see that indeed this is a CD41 low positive cell in the sibling. In the mutants, there are more phosphohistone 3 positive cells, as well as more low GFP expressing cells. So there are more progenitors, and they proliferate more, apparently, in the CHT. Then how about the thymus? In the thymus, there are these lymphoid progenitors. They're homing into the thymus, and there's the definitive wave taking place there. So these, there's a good marker for the thymus and for the thymocytes in the thymus is RAG1. So here's a sibling, here's the thymus in the sibling at 5 days post-fertilization. In the PTEN mutant, the thymus is clearly smaller, as you can see here. And in the close-up of this of the thymus, you can indeed see that the thymus in the mutant is smaller. We determined the surface area, which is clearly smaller in the mutant. So the thymus is smaller in the PTEN mutant. If we then look at what the thymus looks like in this, again in the KDR-like GFP line, you can here in the wild type see the thymus over here. There's some thymocytes moving in and moving out of the thymus as well, but it's a nicely confined area where the thymus is sitting in the siblings. Then in the double mutant, it's a different story. Here's the thymus. It's much smaller. These thymocytes are moving in and out. It's as if they don't know where to home, and they move out and move all over the place around these stages. And so if we take a marker for the for immature hematopoietic cells, CMIB, then we can also detect the thymus over here, as you can see. And in the double mutant, the thymus is still there. There's no difference in size, actually, if you compare it with siblings. In the PTEN mutant, the size is similar of the CMIB positive thymus area, let's say. So this is the picture I've shown you before with the definitively or fully differentiated RAG1 positive cells in the thymus, much smaller, but then the progenitors are still, the size is similar. And there are also some ectopic CMIB, more ectopic CMIB positive cells in the double mutant. So what this all leads to is that our working hypothesis, if you will, there are these long-term hematopoietic stem cells that give rise to short-term hematopoietic stem cells, and they give rise to progenitors and fully differentiated cells. In the absence of PTEN, that's interesting. I mean, 15 minutes, I'm pretty close to time. I mean, that's a bit harsh. What happens in the PTEN mutant is that these, either the long-term or the short-term hematopoietic stem cells, we don't know how to differentiate between the two, they go into apoptosis, so they basically die. And some of the cells, these could be short-term hematopoietic stem cells, they escape, and then these progenitor cells show enhanced proliferation. And these are then immature, not fully differentiated cells. And I've shown you of the myeloid lineage, but also of the lymphoid lineage. And perhaps this enhanced proliferation actually blocks further fully or terminal differentiation of these cells. And so that's the model I would like to leave you with. And then finally, like I said, this was all done by Suma. The work with the hematopoietic stem cells was done in collaboration with Philippe Herbomel, who's also here, and Karima Kissa, who was in Paris but is now in Montpellier. All right, thank you very much.
That was a great talk. We have time for questions. It's great to see you working on hematopoiesis. Well done, you know. Jim.
Yaron, I was wondering if there's a functional consequence to the hyperbranching phenotype. Do they have hemorrhages or other vascular problems that appear?
Not as far as we can tell, at least not in the embryo. So presumably the vessels form, they're pretty much normal. But in the adults, these tumors they form are hemangiosarcomas. But in the embryos, we don't see a functional defect. We don't see bleeding or anything. So, yeah.
Trista.
Hi, that was a great talk. I was just wondering if you, and I don't know exactly how you would look at this, but if the other alternative is perhaps that you're in the CHT and then the result in the thymus is that you've actually expanded one of those intermediate populations of hematopoietic cells and you've actually almost completely lost your HSCs. So then you wouldn't have that lymphoid potential because a lot of those intermediate phases don't. So I don't know how to tell the difference, but it might be another way of looking at it.
Yeah, you might be right, but we also cannot tell the difference. And,
yeah.
As far as we can tell, so with the RUNX1 and CMIP, the HSCs are there, and nothing wrong with them at these earlier stages.
Right, CMIP sometimes compensates, and I didn't know if it would perhaps, you'd have excess production of those myeloid precursor cells from an earlier wave, because it's somehow feedback, and it doesn't know that, or knows that the HSCs are gone. But I don't know how to look at that.
No, right, yeah, so yeah, we maybe naively thought, well, let's try and wipe out the primitive wave using data 1, for instance, but that doesn't really work. I mean, you can wipe it out, but it doesn't really answer this question. Yeah. Yes.
Feng? I do. Oh, good.
Okay.
Tom can wait.
Hi, so I just wondering whether, so you didn't show the PTEN expression pattern, right? I guess it's ubiquitous expressed.
Exactly,
so
yes. PTEN is expressed in every cell.
Yeah, whether the phenotype in hematopoiesis or in vessel phenotype is primary or secondary. The second question is, have you looked at PS3K AKT signaling pathway in the...
Yes, we have. So we can rescue much of the phenotype, at least morphologically, using a PI3 kinase inhibitor.
It's very... Tom.
Haroon, PTEN is very often mutated in thymus-derived TALL in humans. And I was intrigued by your in situ. So the MIB is normal, implying cells are going to the thymus, but then the RAG, how do you interpret the lower RAG again, differentiation?
Yeah, so that's what we think. So we think that differentiation is blocked there. So they don't go to the fully differentiated state, and so they get stuck in a progenitor state, proliferate too much, and do not go all the way to fully differentiated RAG1 positive cells.
But wouldn't you expect to see then an accumulation of MIB positive cells, excess?
Yes, but that's basically what we've shown, although when they are not fully differentiating, they don't stay in place. That's what the movie looks like. I mean, they're there, but then they move out again as well.
So they're moving away.
Yes.
As MIB-positive block
exactly.
cells. Ah, great.
Yeah, I thought it was interesting. So if I've got it right, it's interesting, like for instance, that Sean Morrison has done a knockout of P10, and they have expanded stem cells, at least in adults. But you have more apoptosis in the dorsal aorta, but the MIB staining is normal.
Yes.
So you don't have a molecular marker that actually distinguishes the decreased cell lineage in the aorta.
No, that's right.
But then when the cells get to the CHT, there's a normal number that gets there, or there's less that arrive.
Well, as far as we can tell, there are more cells in the CHT.
Later,
right. So we should probably look at the stage when they start to arrive there.
Right.
Yeah.
We have some markers we'd be happy to share.
Yeah,
It'd be
that would be
interesting.
good. Yeah.
Yes.
I have a question that at the HSC emergency, why the P10 to stimulate the apoptosis at the later stage, they stimulated the proliferation of the cell number.
Right. Yeah. So we don't really know, but that's our observation, right? So early on, P10 is required for the stem cells to survive. If they don't have P10, they die. But later on, and that is actually what we expected, is that if you take out this tumor suppressor, they go haywire. They proliferate and proliferate, and you get more and more and more cells. So.
Okay, thank you very much. That's excellent.
Thanks.
We're going to take a break now. We're going to celebrate that the Bruins have won their 4-3 in this last game, and so we're very excited about that. We'll come back in a few minutes after the break and for our next speakers. So thank you very much.